Assalamu alaikum, dear students. I hope you all are doing well. In collaboration with Scientific Pakistan, we are bringing to you a complete course on NMR spectroscopy. This will be completed in two phases. In the first phase, we will discuss the one-dimensional NMR spectroscopic techniques, and later on in the second phase, we will talk about the two-dimensional NMR techniques. In this introductory session, I will tell you about the course outlines, the literature that you need to study for understanding the technique in more details, and we will discuss the importance of NMR spectroscopy or NMR technique in different fields, especially in chemistry. So let's see what we have today. These are the books that you need to follow to understand the technique in more details. So we have two dedicated NMR books. The first one is Solving Problems with NMR Spectroscopy by Professor Thaur and Professor Muhammad Iqbal Chaudhry. And the second book is One and Two Dimensional NMR Spectroscopy by Professor Thaur. These are two very well-written books. I would recommend all of you to go through these books and understand NMR in more detail. The third book is The Introduction to Spectroscopy by Donald Pavia. Now, this book covers other techniques as well, along with NMR. For example, UV visible spectroscopy, IR spectroscopy, NMR spectroscopy, and mass spectrometry as well. This book has the basic concepts, solved problems, practice problems in all techniques. But the first two are dedicated books for NMR spectroscopy, and I would strongly recommend these books to everyone to read it in more detail. These are the course outlines. So first we will talk about the basic concepts in NMR. Then we will see how we can read proton NMR spectrum and carbon-13 NMR spectrum, because this course is based on one-dimensional proton NMR and carbon-13 NMR techniques. So we will be mostly focusing on proton and carbon-13 for the time being. And later we will see how we can interpret the NMR spectra to get maximum information from these spectra about the structure of a molecule. And finally, we will discuss the application of NMR in structural elucidation. And later on, we will also see some practice problems. We will try to solve these structures with the help of proton and carbon-13 NMR spectroscopic techniques. So what is spectroscopy? Spectroscopy, you all would be aware of, is the study of interaction of light with matter. So when light interacts with matter, and because we're talking about the application of spectroscopy in chemistry and especially in organic chemistry, so spectroscopy here would mean the study of the interaction of light with organic molecules. Why we would like to study the interaction of light with these molecules? That is to investigate the molecular structure and the dynamics of the molecules. Now, this is very important. We have different spectroscopic techniques, for example, UV visible spectroscopy, infrared spectroscopy, NMR spectroscopy, and mass spectrometry. Mass spectrometry is slightly different from the rest of the three because we do not have any electromagnetic radiation used in mass spectrometry, but the rest of the three, UV, IR, and NMR, they utilize a part of the electromagnetic spectrum to allow us to study the changes that occur inside the molecule. So each of these different regions of the electromagnetic spectrum have different frequencies, different energies, different wavelengths, and for that matter, they produce different changes inside the molecule. And those changes are then recorded with the help of these techniques, and the study of those changes help us to investigate the molecular structure or the molecular fragments present inside the molecule. But what about the dynamics of the molecule? Spectroscopy can also help you to study the dynamics of the molecule. You know, these molecules, I always say, they are not dead. They are always moving, they are tumbling, they are changing conformations. So you can study those changes as well with the help of spectroscopy. In other words, stereochemistry can be studied with spectroscopy as well. And as I told you that we have different spectroscopic techniques that help us to elucidate the structure of the molecules. 4 of them are NMR, mass, IR, and UV. These techniques complement each other in elucidating the structure or determining the structures of the molecules. But are they equally important, or are they equally effective in elucidating the structures of the molecules? Let's grade them in their effectiveness in structure elucidation. So the most effective technique of all of these is NMR spectroscopy. NMR gives us information about different types of nuclei present in the molecule, for example, hydrogen and carbon. So these are the 2 mostly studied nuclei in NMR spectroscopy. The study of NMR is not limited to these 2 nuclei only. We can study deuterium NMR, we can study fluorine NMR, we can study nitrogen NMR, phosphorus NMR, but because organic molecules mostly contain carbon in their structure as well as hydrogens, and hydrogen being a very sensitive nucleus in terms of NMR spectroscopy, sensitive means that it can easily be detected in NMR spectroscopy. That is why hydrogen and carbon are mostly studied in NMR spectroscopy. But which isotopes of these 2? Proton and or protium and carbon-13. These are the 2 isotopes of hydrogen and carbon that can be studied in NMR spectroscopy. Now, I have written the effectiveness in terms of stars, so I give 4 stars to NMR because we have 4 techniques. Then we have the mass spectrometry. Mass spectrometry, as I told you earlier, does not utilize any part of the electromagnetic spectrum. In fact, the source of energy is something else in mass spectrometry that we might discuss later on in another course. But it gives us the information about different molecular fragments. And it's slightly less effective than NMR in obtaining information about the structures of the molecules. Then we have the infrared spectroscopy. Infrared spectroscopy is also known as the functional group spectroscopy because it helps us in identification of the different functional groups that are present in the molecule. The information that we gather from infrared spectrum does not give you all of the information that you need about the structure of the molecule. So I give you 2 stars here. And finally, we have the UV visible spectroscopy, which tells you about the presence or absence of conjugation or different types of chromophores present in the molecule, and I give it 1 star. So you can see that NMR is the most effective technique in structural elucidation of organic molecules. What is NMR? Let's try to define NMR spectroscopy or nuclear magnetic resonance spectroscopy. Let's understand this. NMR is the study of the nuclear spins. There are 3 parts of the name: nuclear, magnetic, and resonance. Now, what does these 3 words mean? Nuclear means the nucleus. So we are going to study the nucleus of any atom present in a molecule in NMR spectroscopy. Magnetic means we are going to study the behavior of this nucleus under a magnetic field. And finally, we will observe resonance in these nuclei. So let's see. NMR is the study of the nuclear spins. So we can study nuclei which have some spin under the influence of an applied magnetic field. So we will provide a magnetic field to these nuclei that will bring some changes into the nuclei. Those changes will be recorded, and we will also record the resulting resonance in the nuclear spin through the absorption of radio frequency. What is resonance? You know that when two frequencies match together, a phenomena takes place that we call resonance. So, where are the two frequencies? Of course, one of them is the radio frequency. So, we are providing radio frequency from outside to match with another frequency. Where is the second frequency? That we will discuss in the upcoming session. So, we have three words in the name. Nuclear means the nucleus or the nuclear spin. The study of the behavior of the nuclear spin under the influence of a magnetic field, and later to record the resonance that takes place when we apply or we provide radio frequency to these nuclei. Why NMR is more important? NMR has applications in different fields, but it is more important to chemists. Its application in chemistry is very diverse and very crucial for researchers, especially in organic chemistry and more specifically in natural product chemistry. So it helps us in structure elucidation, that is the determination of the chemical structure of the molecule. And as I told you earlier, you can also study the different dynamic processes that take place within the molecule. When you move a step forward from chemistry and move into biochemistry, where you have these large macromolecules, the biomolecules like proteins, DNA, and polysaccharides, NMR is equally useful in studying the three-dimensional structure of proteins, DNA, and polysaccharides. Another important application of NMR, or the importance of NMR, is drug designing. So we can design drugs with the help of NMR. How is that? You might be familiar with a term known as structure-activity relationship. So if we have a library of molecules and we elucidate their structure with the help of NMR or other techniques, and later on we study the biological importance of those molecules, we may come across a hit or a lead molecule that may ultimately end into a more potent drug. In medicine, NMR is used in MRI. You would also be familiar with MRI, magnetic resonance imaging. MRI is basically NMR. So it's the NMR spectrum of the different tissues that are present in your body. Another important technique that is used in NMR is the saturation transfer difference, or in short we call it the STD NMR, or epitope mapping, that helps you identify the sites within the molecule that may interact with proteins. So the interaction of molecules, organic molecules, with proteins can be studied with STD NMR. So these are some of the important applications of NMR that makes it a very useful technique, and these are the reasons why you should learn NMR. So what are the different steps that we can follow to finally get to the structure of the molecule? Because we are more concerned about elucidating the structure of the molecule, so we will be mostly focusing on structure elucidation in this course. So the first step, of course, is the preparation of sample for analysis. So you have to prepare your sample through an appropriate method to analyze it through different spectroscopic techniques. But because we're talking about NMR, so for NMR, sample preparation is slightly different from others. The first step is to go to mass lab and record the mass spec and the proton NMR spectrum. Mass spec will give you the molecular mass of the compound, and if you have performed the high resolution mass spectrometry, then it might also give you the molecular formula of the compound, and then you record the proton NMR spectrum. Go to the literature, find the molecular formula in the mass spectral libraries. If it's a known compound and you can identify it, you should stop here. But if it's not there and it might be a new compound, then go to the next step. That is to record the carbon-13 NMR spectrum. Carbon-13 spectrum will give you the information about the carbons present in the molecule, and then record the two-dimensional NMR spectra of your molecule. Two-dimensional NMR spectra of different types that we will discuss in another course, not in this one. They may be homonuclear or heteronuclear, giving more information about the structure of the molecule. And when you combine the information, or you combine or connect the dots from all of these spectral data, finally you proceed to structure elucidation and you get to the final structure of the molecule. So rest assured that you're going to have a very unique and interesting journey while studying NMR, because NMR is a very interesting technique and it's very close to my heart because it is interesting and at the same time it's very effective in structural elucidation of organic molecules. So thank you for now. I'll see you in the next lecture, in which we will talk about the basic concepts of NMR and how an NMR signal is generated. Thank you so much.